I, Dr. Sham Zawar, National Mentor for the IVF Laboratories in India, appointed by the Department of Animal Husbandry and Dairying Government of India, on behalf of the Bovine Embryo Transfer Group of India, welcome you all and our esteemed speaker of the day, Professor Pietro Baruselli. Let me introduce our guest speaker. He is Professor of Animal Reproduction at the University of Sao Paulo in Brazil. He did his doctorate in animal reproduction from Sao Paulo University, and then further he was also president of the Brazilian Embryo Transfer Society from 2012 to 2014 and member of Executive Committee of International Congress on Animal Reproduction, ICAR, from 2012 till to date. Professor Baruselli's research and teaching are in the areas of control of follicular dynamics and ovulation for self-appointed artificial insemination and embryo transfer in cattle, both Bos indicus, Bos taurus, as well as in buffaloes. He has over 300 scientific publications and more than 600 abstracts presented in scientific congress on a wide range of subjects, including physiology of reproduction, synchronization of ovulation, then biotechnology of reproduction, reproductive management. You all will be happy to know that till now, 60 candidates have done their master's and doctorate under his noble guidance. He has an extensive international experience as well. Sir, before I request you to deliver a talk, I would like to welcome Professor A.K. Mishra, and we feel proud to have him also as a member of our bovine embryo transfer group. Professor A.K. Mishra earned his bachelor's, master's, and doctorate in animal reproduction from Veterinary College, Mathura. He has recently completed his tenure as the chairman of Agriculture Scientist Recruitment Board, Government of India. Earlier to this, he has served in different capacities, such as Vice Chancellor of the MAFSU, Maharashtra University, Veterinary University at Nagpur, then Vice Chancellor of G.B. Pant University for Agriculture and Technology, Pantnagar. He was also, before that, he was a director of project at the Directorate on Cattle at Meerut of ICR. He has been involved in the development of embryo transfer technology in both cattle and buffaloes. He practiced conventional MOET during the year from 1986 to 2002. He has also established a OPU lab when he was at Pantnagar, and the team performed many OPUs in Sahiwal cows. He has received many awards for his outstanding work. Now, I will also take an opportunity of introducing Dr. Indrajit Singh, who is currently traveling, but he joined for this webinar. Dr. Indrajit Singh is a distinguished researcher, an academician, an educational administrator, heading Gadwasu University as a vice chancellor over there since 2020. Prior to that, he was a director of animal husbandry, Punjab, and he did his doctorate in the subject of animal reproduction from University of Liverpool, UK. He has been invited as a guest speaker on many international conferences around the world. Professor Pietro, as you know, he has been, with his enormous contribution on buffalo research and development, he is popularly known in India as a Murrah man. We call him with a great affection as a Murrah man of the country. Now, sir, I would like to brief you about the committee, a core committee, which is also known as Central Monitoring Unit, which is constituted very recently, just about 2 months back, by the Ministry of Animal Husbandry, Government of India. And I'm heading this committee as a chairman. And I have another 5 colleagues of mine who are the members of this committee, and the main purpose of this committee is to help whatever IVF labs which are spread throughout the country. We would like to share our experience with these labs and help in improving their performance. So one of the member who has not come over here today because of some reasons is one Dr. Mutharao. He is a professor and head at the Veterinary University Livestock Research Station in Andhra Pradesh, where they have got an excellent herd of Angol cattle, to whom you call as a Nelore in Brazil. He has about 30 years of research experience. Again, he is a doctorate in animal reproduction and a good experience in MOET in buffaloes. We have another colleague of ours. You can say hi, Dr. Sanjeev. Dr. Sanjeev Chauvel. He is based in United States. He did his PhD from University of Connecticut, USA. Overall, he has got about 35 years experience, and out of which for the last 20 years, he's been working in USA with a very well-known, renowned embryo transfer IVF company, Transova Genetics. So, and then our third colleague is in the core committee is Dr. Ajay Aswal. He is also a postgraduate in biotechnology from National Dairy Research Institute, having a good experience of 16 years in MOET and IVF. Then Dr. Siddharth Layak, he can say hello, Siddharth. He is a doctorate from NDRI Karnal and a Fulbright fellow from Cornell University. He's working with NDDB for the past 7 years and looking after their ET operations. And we have Dr. Amol Sahare. Hi, Amol Sahare. Amol Sahare, who is also again a doctorate in animal biotechnology, really a good embryologist, having 6 years of experience into the IVF. Last but not least, I'm the, as a chairman of this committee, would like to say 2 lines about myself. I did my doctorate, sir, in the year 1990. And that was the first doctorate in the country on embryo transfer and superovulation in sheep, goat, and cattle. And I have served an organization for 46 years. So I have an experience of about 46 years into this field, starting well back in 1975, when I started my work with the sheep with the Professor Neil Moore. I'm sure you must be knowing from Australia. Sir, with this introduction, now I request Professor Pietro Baruselli to deliver his talk. Thank you very much.
Thank you, Dr. Sim. First of all, I would like to thanks for the invitation. For me, it's a good pleasure to be here discussing with this important research in India. So we have a similar management between Brazil and India. It's a tropical country. We have the same kind of animals, Bos indicus, Bos taurus, and buffaloes. So I think that's very important, this collaboration, this discussion like we are having today with important research in India. So thank you very much for the invitation. It's a pleasure to be here, talking with you, discussing with you biotechnology for production, the results that we have here in Brazil. So thank you very much. So pleasure to be here also with Dr. Misra, Dr. Inderjet, also Dr. Guman from College of Veterinary Science in Ludhiana. Guman is also a good friend that have been here in our lab some years ago. So, and all the research that they are here today to discuss it, this technology that we will present. So I will start my presentation. So just a minute. Dr. C, can you see my presentation?
Yes, sir. Yes, sir. You can,
Okay.
I hope you are put into the full screen mode in the PPT mode, PowerPoint presentation mode.
Uh, is it, it's okay?
Yeah, it's okay, but I think there is a cup down below, and if you put it into a PPT mode, it will be still look a little bigger.
Because in my computer is the full screen.
Okay, sir.
Present.
Okay, okay, sir. Go ahead. Go ahead, please.
Okay, sorry. So, Dr. Sim asked me to talk about the in vitro embryo production efficiency in cattle, compare the study that we did in cattle and in buffalo. So, and as you know, we imported from India several breeds. So we have here Indicus breed and Taurus breed, and buffalo that also we imported from India, Muha and Jafarabad. So we have these animals in our country. So just to know, I work here at São Paulo University. This is São Paulo in the tropics country, part of Brazil. So in our university we have a farm. It's a 2,000 hectare farms. When we have buffaloes, we have cattle, dairy, and beef. And so we are doing the basic study here in this farm, 200 kilometers from here, from São Paulo. But we have a strong collaboration with private farm. So our lab work with the field research that we use farms that collaborate with São Paulo University, and we use also a strong collaboration with the IVF labs that we have in Brazil. So IEDA, WTA, is one of our great collaboration. So we have a strong collaboration with this lab here that we use to produce the embryos and to perform our study in the last 30 years that we are working in São Paulo University. So my lab work with this kind of genetic groups. So we work with beef. So this is the Nelore that we import from India. We have Indicus and Taurus from beef. We have dairy cattle, so we import gear from India several years ago, but we use also Taurus for different breeds and kind. And we imported Muha from India, but we have also Mediterranean from Italy. So we work with this the physiology of reproduction with these animals and biotechnology of reproduction with these animals. So this is our lab. Our lab work with this situation. Of course, we know that the artificial insemination and embryo transfer we use this biotechnology to improve the genetics of the herds and we increase the production. So this is the main topics from using all this biotechnology. So the embryo transfer increased the dissemination or desirable genes, genetic genes, by increasing the number of prints obtained by donors with high genetic values. So we use the best animals here, the best donor, and we use the recipients that is genetically decreasing according to the donors, of course. So using this model here, we improve the genetic of the herd. This is the main why we are using embryo transfer. This just if the embryo transfer is important, I bring you here the numbers of ITS, is the embryo International Embryo Technology Society. So this is the
Professor, I'm sorry, Professor Pietro, disturbing you.
Yeah.
Your slides are not moving, sir. You need to refresh. They're not moving.
Yes, so now it's visual, sir.
Now it's great. Now it's good, sir. Now it's clear.
Okay, sorry for this technology problem. It's okay now?
Yeah, fine, sir. Absolutely fine, sir.
Okay, thank you, doctor.
So we have missed some initial slides, so you can go to them.
Here.
Yeah.
So this is the genetic groups that we are working in our lab, okay? This is the biotech for reproduction. So the genetic improvement and increase of productivity, okay? And this is the main importance of embryo tank technology when we use the best animal, the best genetic of male and female to introduce this genetic in animal with lower genetic evaluation. So, and now is the embryo transfer market in the world according to ITS. So this is important, and we are improving the number of embryo transfer in the world. We are arriving 1.5 million embryo. This is in cattle in the world. But the most important information here is that in 2016 in the world, we start to produce more in vitro embryos than in vivo embryos in cattle. So this is more in vitro than in vivo. So the market is changing in the world. So we are improving the embryo production, yeah, and we are producing more embryo in vitro than in vivo. So look for 2000 here, in 2000, we produce only 10% of the embryo are in vitro. Now in the world, more than 60% of the embryo are in vitro. So it's a rude change in the market in the world. Just to understand Brazil, so this is the number of Brazil from 1995 to today. So we also changed the marketing. This is the IVF in Brazil, but we changed 11 years earlier than in the world. In 2005 in Brazil, we started to produce more IVF embryo than in vivo embryo. And look here, today we produce only 6% of the embryo in Brazil are in vivo. Brazil, we are producing around 700,000 embryos per year and more than 9% in vitro embryo. So this is very important information because we start the new technology early in Brazil. So this is because we introduced in 2000, and IEDA works a lot in this time here. We here start the first IVF company in Brazil in 2000. This private company started to produce a large number of IVF embryos, and also we developed fixed time embryo transfer protocols that improve the number of recipients to receive these IVF embryos. That the IVF, as you know, produce large amount of embryos, and we need recipients that to transfer these embryos. Can program the recipient to receive these embryos. So this is the revolution in our situation. So the IVF today is a reality in Brazil, and we start to work with this more than 15 years ago, okay? So I prepare to present to you some difference in oviral biology response to reproduction technology according to genetic groups. So the ovarian physiology difference that we have here in Brazilian situation between breeds, we use a temporal study between genetic groups, Taurus, Indicus, and Buffalo. the number of follicle, the biomarkers like anti-Mendelian hormones, the antral follicle count, and the oocyte yield. So the in vivo versus in vitro embryo production, impact of genetic groups, and some new technology like a velogenesis, when we are trying to produce here oocytes for prepubertal animals before puberty to reduce the interval between generation and increase the rate of genetic gain. So this is that I would like to discuss with you. If you have some of course, some questions, some suggestions, we are open for discuss with you. So this is just to start. This is a paper that we published in Intelligenology in 1997, trying to understand the follicular dynamics in buffalo, so the ovarian physiology in buffalo. And we found at the time that we have waves, like in works did in the same time in India. So we have a waves, more 2 waves than 3 waves in buffalo. This is the situation, but this we didn't compare with cattle. So what we did 10 years or later, so we put at São Paulo, we introduced in São Paulo University temporal study, comparing Taurus, Indicus, so Holstein, Gyr, and Buffalo, but we use them in the same management, all then are cycling heifers. So we introduce them. This is São Paulo University facility. So we management them with the same nutrition, the same pasture, all then are cycling heifers, 2 years cycling heifers. Look at the buffaloes, the Holstein, the Gyr. So after that, we use one cycle, we synchronize, and we use the ultrasound evaluation to understand the follicular dynamics in these three groups of animals. And we found some difference that is important to discuss with you. First, the estrocyte length, no difference between the animals, around 22 days. But the number of waves per cycle, per Western cycle, is higher in Bos Indicus than in Buffalo and Holstein. So Buffalo and Holstein have the same partner of follicular wave during the Western cycle, but the Bos Indicus, they have more waves during the Western cycle than these other different breeds. The same management, the same situation, the same moment of the evaluation. And when we analyze the corpus luteum dynamics, we found interesting data here. So the corpus luteum is higher in Holstein, intermediate in Indicus, and lower in Buffalo. And when we analyze also the buffalo progesterone during the estrous cycle, the same nutrition, same situation, they have lower progesterone comparing to Indicus and Taurus, and they have lower corpus luteum also compared to Indicus and Taurus. So this is difference. Of course, when we use a buffalo as embryo recipient, of course, we will have a smaller corpus luteum comparing to the other breed. But this is a typical physiology from buffalo. They have a high fertility with this kind of response, but we need to know the difference between these species. So we study also the follicular deviation. We published this. We will not have time to discuss with you, but we found in buffalo, this is the FSH, LH, the moment of deviation. It's some difference between species. When we use all this paper that we publish, compare also with Ginter in the United States. So the buffalo achieved the deviation in 7.2 millimeters, the Taurus 8.2, and the Indicus 6.2. So this difference, we know about that in follicular dynamics. We need to know this difference, but the most important difference in physiology, I will start now to discuss with you, is the number of recruited follicle per AVE. Because when we are talking about embryo transfer, of course, we need to retrieve a large amount of sites. So we try in our lab to study the difference between these species according to the number of follicles present in the ovary. So also in the same farm in São Paulo University, we use the same model, temporal study. We use also, again, other heifers, so 2 years heifers according to cyclicity, Indicus, Taurus, and Buffalo in the same management, same situation. And we synchronize the wave using progesterone and estradiol. As you know, we have a beginning of the wave 4 days after the treatment. So here we can have more than 9% of the animals starting the follicular wave. So at the moment here, we did the evaluation to understand the number of follicles according to the genetic groups. And look here, this is a very important information. When we use IVF or biotechnology for reproduction, Bos indicus, the donors, they normally have the double of follicle compared to Buffalo and Holstein. So Buffalo and Holstein, they have very similar number of follicle in the ovary, but the Bos indicus, they have a high number of the follicle. So produce IVF embryo in Bos indicus is more efficient than Bos Taur and Buffalo because they have high number of follicles in the ovary. This, you will see that we did the replication of this information in temporal study and have a high repeatability of this information. So more than 90% of the studies showing that Bos indicus present more follicle in the ovary than Taurus and Buffalo. So this is important. This is another study we did. This is after synchronization. This is after ovulation in normal estrus cycle. So 2 days after the ovulation, after the estrus, we did the evaluation to understand the number of follicles recruited at the beginning of the first wave. And look here, again, here is gear. Gear producing more follicle at the beginning of wave compared to Holstein and Buffalo. And Holstein and Buffalo the same amount, and the Zebu the double of number of follicles. This is the first ovulation. This is the second ovulation of estrus cycle. After the first ovulation, after the second ovulation, look that we have a high repeatability of this information. So normally Bos indicus is present a large number of follicles compared to Buffalo and Holstein. So is the AMH a marker, endocrine marker for number of follicles? Because we need to select the animals as donor. Né? As you know, more follicle is correlated with more IVF embryos. So we try to study biomarkers for follicle, antral follicle count and for IVF. So we did again a temporal study, and we published this study trying to understand the correlation between this biomarker hormone and the number of follicles. And look that again, when we analyze the amount of AMH in black here is the amount of blood AMH, circulating AMH, is double, 3 times higher in Indicus compared to Holstein and Buffalo. Holstein and Buffalo, no difference, statistical difference between antral follicle population and IMH, but again, Bos Indicus have a higher IMH and higher antral follicle population when we analyze in temporal study. And in the 3 breeds, the IMH is correlated, high correlated with number of follicles, independent of the number of the concentration. When we did the correlation between IMH and number of follicle, the IMH is be a biomarker for a number of follicle in the ovary. We did several studies that showing this high correlation. So just to understand the efficiency of in vivo versus in vitro, mainly in buffalo, I will introduce you some research that we did trying to understand the efficiency of in vivo and in vitro, mainly in buffalo, because when you use cattle, this is Gabriel Boas studies with Ruben Mapletop. They have a strong work in trying to understand in cattle the in vivo embryo production, but in cattle, when we did a flush, we produce in mean 5 to 6 embryos. Of course, this is high variable. We can have a 10 or we can have a 2, but the mean is 5 to 6 embryos per flush. But when we introduce the study in buffalo, this is a problem. This is, you remember your works, of course, sometime we can produce some number of in vivo embryos, but when we analyze the literature, the study of Martin Dross, Professor Misra, Professor Ziccarelli in Italy, our study here in Brazil, the embryo production is lower in buffalo, in vivo embryo production compared to cattle. So does the buffalo respond to superioratory treatments? So just to show, Misra have an elegant study, but I will show you the study that we did some years ago. This is a study that we did in 1994 with ultrasonographic analysis of superovulation animals. And so what we found that when we synchronize a buffalo, so here is the day of the first FSH, the day 10 of estrous cycle. So we have a dominant follicle. We have at the beginning of follicular wave in the second wave, buffalo second wave. This is the recruited follicle at day 10 of estrous cycle. So we start the FSH. Look that this is in the ultrasound that we use. This is in 1994. The quality is not so good, but look that they start to respond to superovulation. This is 24 hours after, more follicles is growing to after FSH, follicle is growing. Look at large amount of follicle in both ovary, left and right ovary. So they are responding to FSH. Look here, this is the day of estrus when we are doing AI after estrus detection. Now we can synchronize, but at the time we use estrus detection. So look that they respond. So they ovulated. This is 24 hours after. They are ovulating. They are ovulating. So they produce, this is the corpus luteum that they produce in the day of the flushing. So we did this analysis in large number of buffaloes, superovulated buffalo, and they respond, they have a good response to superovulation. But are the embryos into the uterus? This probably will be the problem. So we did a post-mortem embryo transfer analysis in superovulated buffalo. So we superovulated them. This is in São Paulo University. We have a slaughterhouse. So we superovulate the donor and slaughter the donor, and we collect the genital system. And look here, large amount of corpus luteum after superovulation. And we did the flushing of the oviducts, flushing of the horns, uterine horns, and we measure the fimbria, and we try to understand what's happened with the superovulated buffalo, because they respond with corpus luteum. But look here, this probably is what's happened. They have 90 corpus luteum, but we flush after post-mortem. We collect only 3 embryo. Is oocyte and embryo. It's not embryo. Oocyte embryo. Only 35% of the corpus luteum is correlated with embryo, oocyte of an embryo collecting the uterus. As you know, if in the literature, this correlation in cattle is around 6 to 8%. So probably here is the problem. They respond, but when we flush the uterus, we are not collecting the embryos. So where are the buffalo embryos after superovulation? We don't know. We did a lot of study. Julia Soares will talk with you about a sequence of study we did in our lab, trying to understand the problem of in vivo embryo transfer. But Julia will talk about it. So because that we start years ago to produce in vitro embryo in buffalo, and we try to compare the efficiency between buffalo and indicus and taurus also. And so the effect of genetic group in the follicular population, in IVF production. So I will start now to show you the study that we did comparing these 3 genetic groups for IVF, in the same condition, in same situation. So this is the study for Lindsay Jimenez. She did a master and PhD with me. Today she's a professor at São Paulo University. And so we compare in the same situation, indicus, taurus, and then buffalo. So Lindsay did in the same lab. She did all the analysis. We collect the oocyte in the same day. We produce the embryo in the same conditions, but our model tried to understand the effect of the breeds, the genetic of the animals. And see here, this is the one data that we did some years ago or so. But look at the Nelore, the number of follicles in green. Look at that buffalo have the same number of follicles, but the Nelore, the double follicle. The oocyte recovery after OPU, the same, the buffalo and Holstein around 15, but look at the bull, 37. Here, cleaved embryos, look at the buffalo and Holstein around 5 embryos, but the bull 15 cleaved embryos. In blastocyst production, so here 5 embryos and here 1 embryo. So this is a strong effect of the genetic on the embryo production in the same situation, the same lab, in the same group of the research. So look that we can produce embryo for Holstein in buffalo, of course, but Bos indicus is higher embryo, in vitro embryo production than these other genetic groups. Of course, we have a strong variation between donors, of course, but normally Bos indicus produce a high amount of IVF embryos. So just to understand, here we did a study only comparing gear that we import from India and hosting, the same situation. I will show you that is not only amount of oocyte, but the quality of the oocyte from Bos indicus from IVF lab. So what we did, we used a temporal study. That this is an area that did a PhD study with me in 2010. So we use the same model, the same labs, the same nutrition for gear donors or for hosting donors. And we produce the oocyte retrieval, look more in, again, more in gear than in hosting, viable oocyte, more in gear than in hosting, cleaved oocyte, more in gear than in hosting, blastocyst yield. Look that we produce 5.4 times more embryo when we use a indicus donor than a Taurus donor. Again, the gear, the zebu, again, in another study, producing more embryos per IVF. Look, you use the same lab, the same semen, the same condition in this analysis that we are showing you. And it's not only quantity. Look at the analysis that we are doing, trying to understand the quantity and the quality of the oocyte. So look here, the viable oocyte rate is higher in indicus than in Taurus. When you use the cleavage rate, is high in indicus than in Taurus. And we use blastocyst rate, is high in indicus than in Taurus. So it's not only the quantity, but also the quality. So in our Brazilian situation, when you use an indicus as a donor, it's a great quantity and better quality of retrieved oocyte compared to Taurus. And normally Buffalo have, in our study, is more similar from a hosting than a gear or a Nelore or a indicus donor, okay? So if this is great quantity and better quality of sites in indicus, this is important information because when we start the IVF in Brazil in 2000, and as I show you the data, in 2000, Brazil started to produce high amount of IVF, and 90% of the market at the time in 2000 is from indicus, because in 2000 have a strong value of indicus genetic in Brazil. So probably the IVF embryo improve a lot quickly than in other part of the world, because we start the industry producing zebu indicus embryo. So it's more efficient and support development of IVF. Now Brazil is changing for dairy, for Taurus, but at the beginning, 90% of the embryo we are producing 2000 is indicus embryo. As you show, indicus have a high efficiency in embryo production compared to Taurus, okay? So this endocrinal marker we use for the amount of follicle, but we try to correlate the endocrine marker for IVF also in these three kind of genetic groups. So the AMH, as you know, we have in the literature Ireland study, Professor Monio study, Michael Doctor also did some study, and others in the literature. So the IMH is a marker for antral follicle population, of course. We know by the literature, and it's correlated with supraovulation. Alexandre Souza did some study, Rico did some study, Monio. So we can correlate the concentration with MH is a positive correlated with the number of embryo produced after the in vivo superovulation. Okay, so what do we try? We try to understand in tropical conditions the relation between IMH, follicle population, as I show you, but also using IVF. So the IMH is a marker for select IVF donors in this breed. So this is a study that we published, if you'd like to see in the literature, but we found great correlated with AMH in Taurus and in Indicus. So look for the Indicus data that we presented in the literature. This is the number of donors. We use 34 donors in this study. The high correlation with AMH and follicular population in viable oocyte retrieved, in viable embryos. So it's a marker for amount of embryo, IVF embryo produced. But we found here that is not a marker for quality, it's a marker for quantity. But when we use viable oocyte rate or blastocyst rate, the AMH is not correlated with quality, but it's correlated with quantity of IVF production. This is in hosting. We did the same in hosting. Look here, we used 59 donors in the study. We measured the AMH of the donor at the moment of PU. High correlation, follicle population, viable site, viable embryos production. But again, no correlation with quality. So IMH is correlated with quantity, but blastocyst rate, no correlation. And in buffalo, we did the same in buffalo. We used the donors, buffalo donors. We measure in 72 buffalo donors. We measure the amount of circulating IMH. Look, again, another study, high correlation in buffalo, IMH and follicular population, viable site, and viable embryo. So it's a biomarkers for quantity, but it's not a biomarker for quality. Look that again, when we use the viable site rate or blastocyst rate that we measure for understand the quality of the embryo, no effect, no statistical effect using AMH in buffalo also, in buffalo donors, using the AMH as a biomarker. It's a biomarker for quantity, but it's not a biomarker for quality. And so we did another question, and the fertility of the embryo produced according to IMH or according to amount of the follicles. So we did a study, we measured the AMH in the donor, and we transferred the embryo in recipient, and we analyzed the probability of pregnancy according to IMH, and look that no effect in the fertility. So if the donor have a high IMH or low IMH, the conception rate of the embryo is the same. This is a study that we did in hosting. So IMH is not correlated with pregnancy rate. And here's another study that we did also in 4,000 hosting. Look at the amount of the probability of pregnancy according to the number of antral follicle population. So here we expect to have a high IMH and low IMH, no effect on the fertility after 4,000 embryo transfer. So no effect. Here in Bos Indicus, 3,000 embryo transfer, again, high amount of antral follicle population. So we start to see a little decrease in the conception rate in the animals that we have more follicle, more antral follicle population. So no positive effect, and probably a little negative effect of amount of follicle in the ovary and conception rate of the embryo production. So this is for a better study. This is in buffalo. We did also a study in buffalo, trying to understand the probability of blastocyst peropiu. Of course, selecting animal with more oocyte, we improve the blastocyst peropiu, of course. But when the blastocyst rate, no effect, and the probability of pregnancy of the buffalo embryo according to antral follicle population or IMH is not positive, and we have also a negative effect like I show you in Bosnia. So at the end, to finish the biomarkers, hormone biomarkers, or to correlate then with IVF production. So IMH and follicle population is positive. IMH and number of embryos is positive, but IMH related to oocyte quality and related to fertility is not positive. So look that we have this situation according to the IMH as a biomarker. But this is another study that we did in Taurus and in Indicus and Buffalo. So the repeatability of the number of follicle in the follicular waves. So we know that we have animals with high antral follicle population, low antral follicle population within the breeds. So it's important for IVF select the donors that recruit a large number of follicles, because this is the information that they have a high repeatability. So when we use this model in Bos Indicus, we did a study using 36 donors, the same donors we collect for 12 months or 1 year, once a year. So we produce to understand the repeatability of the embryo production according to the donor. The same seeding, the same model. You look here, high repeatability. If you select a donor that produces a large amount of embryos in the first of you, they probably have a high probability to produce high number of embryos at the end of the program. So select animals for number of retrieved follicles is very important for the efficiency. So we published this data. Look here, if we select the animal, here is the program. After 3 months of work, 6 months or 12 months, the same donor. So if we select the animal with low number of oocyte recovery or high number of oocyte recovery, we can produce in the same time, in the same management and working, we produce the double of embryos selecting animal according to the follicle population or number of retrieved products. So this is important to the efficiency of the program. And in buffalo, we know also, buffalo present the number of follicle or site retrieved very similar than hosting, but we have this variation. So we can select the buffalo according to the oocyte retrieved. We have, of course, the buffalo need to have a high genetics and large number of follicles. And if we select this buffalo here, we improve significantly the efficiency of our IVF system, okay? And now we would like to discuss you new technology, like the technology to select the most genetically efficient donor. So the question is, which is the most efficient donor? If you analyze the one genetic program, the most efficient donor is these young animals, because this is a new generation. The genes that we have from a genetic program are more present, the positive genes is present in the young generation and the old generation. So we need to try to start now our program using the young animals. Of course, the young donor sites using in correlation with young bulls reduce the generation interval and increase the rate of genetic gain. So this is the philosophy of the program that we are introducing here in Brazil today. Of course, we use the genome analysis of these young animals. If they have a positive genetic and genomic analysis, we are starting to produce embryo, IVF embryos in young animals. It's the same in buffalo. The donor, the best donor, the best genetic is here, is not here. Of course, if this donor is good, the next generation is better than the old generation. This is the concept of the program. And for produce embryo for young donors, what we can understand also is the accuracy, is the genetic gain is related to generation interval. And if you decrease the generation interval, we can improve the genetic gain of our program. And so this is not new. This if you can see in the literature, the velogenesis is a technology that was recognized more than 60 years ago. It started this technology. So just to understand better, so if you use old donors, the interval between generation is long, like 3 years. But if we use a young donor, we decrease the interval between generation in 1 year. If we imprint, like example, 1,000 liters per generation, so here we need 15 years in the program, use 1,000 liters per generation to arrive to 5,000 to 9,000 liters per lactation. But using the velogenesis or the technology to use calves as a donor, we can reduce here the time when you can arrive at the same production, but 10 years, reducing 10 years the genetic program. So this is we are studying a lot this in Brazil today, trying to understand. And this is the potential for IVF in young animals. This is Brahma. This is in Australia, Michael Dockel, that we have a strong collaboration. Just to show the potential. This is the ovary from a 3 months Brahma. This ovary here has 100 follicles after FSH FSH treatment. So look that we have a great possibility to produce large amount of embryos using this technology in our situation. So this is the study that we did in São Paulo University. This is Bos Indicus, 2 months old donors. Look here, the ovary, supraovulate ovary, using laparoscopy to collect the oocytes. So we did this study in Taurus. This is calf, 2 months to 4 months calf. This is the Bos Indicus calves. And this is the laparoscopy we did to collect the oocyte. So see here, this is the cycling, adult cycling heifer as a positive control. But look that in calves treated with FSH, you can collect, look at 32 oocytes per laparoscopy. It's a high amount. Of course, the conversion, they decrease the rate of the comparing to cycling to adult animals, But look, we can produce around 2 embryos. In this study, we produce 2 embryos per calves, lower than cycling heifer, but showing that's possible to produce in calves. This is the hosting. You look here, we collect 54 embryos per hosting, 12 calves. We produce lower amount of embryos, of course, than cycling animals. But when we transfer the embryo, this is the conception rate. This is the calves born. The mother, the donors, was 2 months, and this is the calf. When we produce the embryo, the mother was 2 months. This is the conception rate for adults and for calves. It's showing that's possible. We are trying to improve the efficiency, but this is showing that's possible. This is the calves also, the Taurus. The mother was also 2 months. 2 months, and now they are producing. And what is the problem for the cow? We don't have access for the ovary by ultrasound. So we did the AMH as a biomarker for cows. And look up, the AMH in follicular population and numeroocyte recovery and embryo produced for cows is higher for Holstein and is higher for also for Zebu indicus. So the AMH could be a marker for select calves as a donor, because we cannot, we don't have a lot of information for the number of sites or number of follicle retrieved. Like in adult animals, you can use the ultrasound to select, but in calves is more difficult, so we can use AMH as a marker. Okay, and this is the buffalo. We did the same. This is the buffalo donor that we use in this experiment. Here's the laparoscopic buffalo. They are also 2 to 4 months. So we use also adult animals as a control using OPU. And you can see here, you produce for calves 1 embryo for buffalo calves. And of course, the adult animals produce a little more. But when we transferred 80 embryo produced for calves, we got 37% pregnancy rate. That's similar than adult donors that we use in our experiment. And this buffalo here, they born at São Paulo University, but they have here, the mother. When we produce, this is the mother. The mother, the donor was that, and the cow was born was that. So now they are, we introduce them in the system. We have at São Paulo University in the farm, a group of Murra buffaloes that we are using as for research and milk production. Okay, and just to finish, we are studying with IEDA and WTA also. We are studying a probe, a follicular aspiration guide, and for young animals. So this diameter is reduced, and here Bernardo is aspirating not more 2-4 months, but 4-8 months calves, but using laparoscopy and OPU. Look here have also young animals, 3 to 5 months. We are doing aspiration using today this probe that have a lower diameter than the conventional probe, trying to understand. And here in gear, we aspirated this donor here 3 months, 7 months, and this cow was here born from these donors using not laparoscopy but OPU, normal OPU, trying to reduce the interval between generations. So this is the the philosophy of the program, trying to introduce technology to improve the efficiency of the genetic program. So we are here decreasing the interval between generations. This donor here, all of them have a genomic evaluation. They are part of a Brazilian program for genetic evaluation. So the farmer did a genetic evaluation, a genomic evaluation, select the best cows, we produce embryo and transfer in recipient, and of course, the new generation arriving in 1 year, 1.2 years. So this is important to accelerate the genetic program of the animals. So as a conclusion, the endocrine and physiological difference between bovine and buffalo reveal a number of key points related to manipulation for the ovarian follicle growth. It must make to account to improve the efficient assisted technology in this species. I show you that that the IVF is used around the world, and the indicus generally produce more oocyte, certo, than taurus and buffalo. This is important to understand the efficiency of the IVF program. So I think that all this information here is important for the industry, for the IVF program, for the genetic programs. You need to understand the difference between the genetic groups, trying to improve the efficiency of the system. Of course, we need to study a lot of trying to improve more the efficiency, but this is the information we have today. And so if someone would like to have this information, so this is a study, a review that we published in 2020, and we introduce here all the information regarding to embryo transfer in buffalo. Part of them I show today for you, but in this review here you can have the details of the information. I think that if someone would like to see the more the information, I suggest you to take this study in account. Okay, so is that so? Thanks. Great pleasure to be here, to see all the friends. Then again, Dr. Inderjet, Dr. Misra, Dr. Guman, and all the people who are present. Dr. Sam, thank you very much for the invitation. It's a great pleasure. I would like to acknowledge my graduate student. So I present today several works did from them. Julia will talk with you in the next meeting. She will present more the information from buffalo superovulation. Julia, Dr. Nelson, so they did several works in collaboration during the master and PhD. Now they are doing an elegant study in the research institute, the Buffalo Research Institute that we have here in São Paulo State. Okay, so is that. Thank you very much. So if you'd like to discuss some details that I present to you, it's a pleasure. So, and I pass for Dr. Sian the words.
Thank you, sir. Thank you very much, sir, for such a illustrious and talk of yours, which really covered so many research projects in such a short span of 16 minutes. And you took us through all the differences between the Indicus and Taurus and the buffaloes. And I remember meeting you during the SBT meeting and many times on the IETS meetings. Always have been a great pleasure in meeting you and talking to you. And I could see Bernardo doing OPU and myself and one of my colleague member who is there, right now in this meeting, Dr. Vinod Patil, we had an opportunity to see Bernardo doing the OPU of the Murrah Buffaloes, where Lucia and Elcio were there. And taking some clues from there, honestly, we came down to India and immediately started a trial in buffaloes, and we succeeded in production of the IVF calves at the doorstep of the farmers for the first time in India. And we also repeated subsequently with another breed of buffalo in Gujarat known as Bunny. Sir, your talk has been so, it will be inspirational to all of us. And in context to buffaloes, we have a tremendous work and a scope in India to do it. And we definitely look forward for your continuous guidance. And we will also request you if you can kindly help us in collaborating with some of our universities in jointly taking some buffalo work. I can say, because here Dr. Indrajit is here and his team is also there, and Ludhiana, as you know it, and they have nice IVF laboratory over there. So that one, and another one is that National Dairy Development Board, wherein also they have recently started good work on IVF. So like that, we can shortlist about 2-3 institutes, and in collaboration with the Sao Paulo University under your guidance, we can take up this technology further, and really it will be a great help from you to us in not only in buffaloes, but in the other cattle breeds also, Indicus and Taurus. Before we move to the question and answer session, I will now request Professor A.K. Mishra to throw his views and some light on this whole thing. And subsequently, I'll request also Dr. Indrajit Singh and my committee members if they have to say a few words. And subsequently, sir, we will look into the question and answers. And thank you very much once again. So over to Professor Mishra.
Thank you, Dr. Zawar. Organizer of today's webinar, Dr. Zawar, the National Mentor of IVF Labs of the Department of Animal Husbandry and Dairying. Professor Pietro Baruselli, the very distinguished speaker of this webinar. Dr. Indrajit Singh, the Vice Chancellor of the Gadwasu, Ludhiana. Dr. Ghuman, Dr. Chobal, and all my colleagues and friends of Bovine ET Group. First of all, let me thank Dr. Zawar for creating this group, which has certainly become an excellent platform for learning from each other's experiences. That includes overseas experts also, like Dr. Baruselli and many others. I would like to compliment first to Dr. Baruselli for his excellent presentation, having many meaningful insights related to the ovum pickup and IVF technology. And I'm sure the participants must have benefited because a comparison was made between Bos Indicus, Bos Taurus, and Buffalo. We are working on all these breeds. And particularly, we are in the initial stages of development in Bos Indicus and Buffalo. Though we started, as Dr. Zawar introduced, I could set up a lab in 2002 to work on buffalo and indigenous animals. That was a project funded by the Department of Biotechnology. And I think we must have performed about 1,000 OPUs in Murrah buffalo and Sahiwal cows. One of my students, Dr. S.K. Yadav, is in this group. He must have performed 300 OPUs in buffalo. And that lab subsequently, I think in 2012, not 2008, produced the first buffalo calf using OPU IVF technology. This technology, I personally feel, is very, very important. And I am so glad that Government of India has started giving a lot of attention to the livestock sector, particularly the dairy. We all know that the government has established a separate department with a full cabinet minister to look after the animal husbandry sector. And it is so heartening that this year's even budget of livestock sector has been increased by the tune of 40%. If we see the whole dairy sector, our average productivity is very low. And when we go for identification of bulls, either for frozen semen stations or for natural service, they are available in very, very limited numbers. And they are sometimes also not meeting the requirement. So therefore, we have to have a technology which can solve our problem. And I consider this embryo transfer as one of the disruptive technologies that can revolutionize a dairy sector. Though we started the whole program of embryo transfer as early as in 1986, we established technology, but it is unfortunate that we could not use this technology as a national program for the livestock improvement, particularly cattle and buffalo. And it is so heartening that now, of course, under the Rashtriya Gokul Mission, the Government of India has established as many as, I think, about 31 laboratories. And there are another about 10 laboratories which are working in private sector or in universities or ICR institutes. It is very, very heartening. And when I see the results which are emerging either from Dr. Zavar's group, Dr. Chaubal's group, and many other groups, sometimes I keep reading the messages which are coming in this particular our WhatsApp group. It is very heartening. Some of the donors has produced more than 100 calves. So this can certainly revolutionize the sector. And Dr. Zavar indicated that he has initiated the work on buffalo. And the talk of Dr. Baruselli has indicated that certainly we can recover a good number of oocytes from buffalo also. I completely agree with Dr. Baruselli when he was saying buffaloes do superpopulate. During 1994 or 95, during my PhD, I sacrificed 18 buffaloes after superpopulation. And I collected genitalia and I observed from my naked eye. The number of ovulations were almost 12. I'm talking of average. That is a reasonably good number. But only problem is we are losing embryos. I also could not recover more than 40% embryos. Initially, there was problem when we were collecting embryos non-surgically. We were collecting at day 7. But this experiment indicated that the buffalo embryo development is at least 24 to 36 hours faster than cattle. And buffalo embryos, they come into uterus around day 4.5, and they invariably hatch on day 6. And that we could correlate with our earlier observations when we flushed large number of buffaloes. That was the presentation made by Dr. Kurup in World Buffalo Congress. Our total embryo recovery was 1.1 or 1.2, and we recovered less than 1 viable embryo. But we were recovering many ghost Jonah pellucida. We could not understand that. But when we sacrificed buffaloes, we realized that we have to advance our non-surgical collection. And after that, we started recovering good number of embryos, I think about 3. But if that work can continue, I'm sure our recovery and superpopulation will improve with a better understanding of the physiology of buffalo. So that is one. And but today, Dr. Baruselli has indicated that yes, this ovum pickup and IVF is successful in buffalo. I was only thinking that his recovery, particularly in Bos Taurus, is a poor as compared to other countries which are reporting as many as an average, the blastocyst development of about 6 per pickup. That's fairly good, fairly large, I will say. And in India also, I think our colleagues are reporting development of blastocysts to the tune of 4 to 5 per collection. I was talking to Dr. Chaubal also. So that can be considered to be very good. Now here, another good thing which Dr. Baruselli has demonstrated is our Bos indicus is far better than Bos Taurus. That's a really good silver line. Because of the crossbreeding, we have lost many important breeds. Now, when we tell our farmers that, you go for indigenous breeds, they immediately say, you make us available indigenous breeds. And those animals are really not available. If you see Gir, they are in sizable number, but again, when you go for a farm, you select the best genetics. So that best genetics is not available, and this technology can certainly help in multiplying this, whatever superior animals are there in a very, very faster way. The second thing which I would like to talk is, as our labs, they start producing more and more number of blastocysts, more number of embryos, how to channelize it for transfer and production of more number of calves, particularly at farmers' field. I would like to share with Dr. Zavar, I happened to visit, Holland sometimes back, I think almost 10 years back, more than that. There I saw they have a system, it's a very small country. They have a central IVF laboratory where IVF work goes on. And to that laboratory, there are 3 ovum pickup centers attached in 3 corners of the Holland. So on these centers, they simply do the ovum pickup, nothing else. Within few hours, they harvest the oocytes. Whatever good oocytes are there, they are transferred into maturation medium and they are shipped in a CO2, portable CO2 incubator to the central laboratory. There further processing goes on, and when the embryos are developed, they are shipped back in the same direction, and then these embryos are transferred into recipients. So I think some sort of system we have to develop in our country also, because our country is a very big country. Even these 30, 40 laboratories are just not sufficient. So some, I think, initiative you may take as a mentor of IVF labs. This will also ease the problem of recipients. And ultimately, I think no farm can maintain. It's a very costly affair to maintain recipients. If we have to take this technology to the farmers, if we have to make use of the animals which are available in the field, I think this will be a very good option. I consider this again to make a revolution in the days to come, and it can ease our problem of availability of elite animals, particularly the breeding bulls. I have, I think, one or two queries with Dr. Baruselli, which I can flag. It was very interesting to know that this anti-Mullerian hormone is an excellent marker to judge the availability of follicles which can be recruited. But I was just looking for some work where you have manipulated this hormone and seen the result. Can we really manipulate it? Can we increase it or decrease it and see the effect on number of ovulations which are happening or number of follicles which are recruited? This will be one of my query. And then I think time is not there to talk more. I would like to thank Dr. Zavar again for organizing this webinar and giving me this opportunity to talk to all of you, and particularly to the Pietro. After a very, very long time, I'm able to see him. Thank you very much.
Thank you. Thank you, Dr. Misra. And before I request Dr. Indrajit Singh, I would like to just add a few things on the points which you told about your experience of Holland. Let me, update you on this, and as well as Professor Pietro, that in the last 6 years, when I was working with JK Trust, for the IVF, we did all these sort of experiments and very successfully bringing oocytes from all corners of India and bringing it to our lab near Pune, and traveling back the embryos across right from Kashmir to Kanyakumari, I can say, east to west and north to south. So we did all that, and we could establish pregnancies at various places. Second very important experiment, Professor Vitro, which we did, is that to get a confidence of the IVF technology at the doorstep of the farmers, we had about 4 IVF mobile labs, which are fully equipped, a very small van, but fully equipped with all the equipments, and where we park that van at the doorstep of a progressive farmer, collect the oocytes and produce the embryos in that lab at his farm itself into the van, and then transfer it. That really created a lot of interest into the farmers, because our farmers, they really got a confidence as to what this technology, that instead of taking the oocyte to your own lab and then bringing the embryos to spread this technology, we did it. Another very interesting work which was done is that hiring the wombs, some of the farmers crossbred, the HF crossbred recipients, they were having it, and they don't want to rear a heifer calf. So we were hiring the wombs like in human beings. It is there some practice in some part of India. So we were hiring the wombs and paying those farmers 10,000 rupees. And when the calf is born, which is 4 days old calf, we were bringing that IVF calf to our farm and rearing it for production of the good bull calves for the semen station, as rightly said by Dr. Misra. And you have a tremendous contribution in the field of cattle and buffalo, as I know you for now almost about 4 decades. So we appreciate your contribution in this field. Now I request Dr. Indrajit Singh to put few words about his experience, and then we will switch to others. Thank you.
Thank you, Dr. Shyam Jawar. And first of all, I must compliment you for organizing this series of lectures by eminent speakers, including Professor Baruselli, which is really, you are acting like a mentor for our embryo transfer people. And many of our young researchers in embryo transfer technology, they are attending this seminar. And I don't want to be between them and Professor Pietro. So I don't want to say much, but I'm really delighted to see Pietro after a long time. And there is no doubt that he is the most versatile researcher in embryo biotechnology or reproduction, particularly in buffaloes and zebu cattle. And whatsoever he has told in this presentation, I think definitely our young researchers, they will get benefit from that. And it was nice to see Professor Mishra as well, who has been pioneer in embryo transfer technology in this country, starting from National Dairy Development Board to GB Pantnagar University. And he is also a mentor to several people who are engaged in this technology. So I think it is very heartening to note that indigenous buffaloes or Bos indicus, they are the best responders to OM pickup. And in case buffalo is performing as good as Bos taurus, then I think still this technology is applicable to buffalo as well. And that has also been shown recently by Dr. Jamar and his team in both Murrah buffalo as well as in Bunny buffaloes. So I wish all the best to these seminars, and thanks a lot, Professor Pietro Baruselli and Dr. Mishra and Dr. Shyam Jawar for giving me this opportunity. I couldn't see to the slides because I was driving, but I could hear all around, and I was well within time at my destination. Thank you very much.
Thank you, Dr. Indrajit Singh. Now, I just quickly wanted to request our core committee members if they have to add something. I just request Dr.
Just one point.
Yeah, sorry. Yeah.
You know, I just wanted to say you have an excellent infrastructure in Raymond's. You are doing excellent job. But my concern was, you know, centers attached to other ET labs which are coming up. Because everybody will face problem of recipient.
No, sir,
That was my point.
absolutely.
I don't doubt the technology.
No. I appreciate your suggestion, but I just wanted to update you on this. Now I'm no more working with Raymond also, and I've been fully devoted myself to as a national mentor for all the IVF labs in the country. And with my colleagues as a core committee members who are there with me, we will try to help these labs and try to solve their problems. Really, it's not a regulatory body, but we wanted to help them in improving their results and get the maximum blastocyst per OPU and the maximum pregnancies at the maximum calves. Ultimately, with that objective, we are here to help all the IVF labs of the country. And that's the whole objective and a motto of this CMU as well. So I request Sanjeev Chaubal, who is from USA right now, from Transova, to add a few comments. Please, Sanjeev.
Okay, thank you, sir. First of all, thanks, Dr. Baruselli, for an excellent presentation. I was very happy to see Dr. Mishra also after a long time. He is my mentor. And again, a lot of friends and colleagues. I have been fortunate to work in India in this field for almost 15 years, and then in USA for last almost 20 years. So I would say that I have seen both sides of the world, and I have been fortunate to visit Brazil also and have been interacting with some Brazilians. So I know what is going on there also. So commenting on these three countries, I would say that in my opinion, Brazil is probably 15 to 20 years ahead of India, and USA is probably 30 to 40 years ahead of India. So I'm not interested in catching up with USA. I'm more interested in catching up with Brazil. And so my dream is, or my thought is, can India be the next Brazil as far as cattle IVF is concerned? Because our conditions are more or less similar. And what Brazil has done is just a very good example for India to follow. I would go back to Dr. Baruselli's very early comment when he said that why in Brazil IVF embryos started overtaking in vivo produced embryos. And he pointed out two main reasons. One was starting of commercial companies to do IVF work. And second one was starting fixed time embryo transfer. That means they started giving importance to creating an infrastructure for large scale embryo transfers. As Dr. Mishra also has pointed out, and we have seen over the last few years in India, most of the labs that are working in India, they can produce embryos. Embryo production is not an issue. The main issue is transferring those embryos and getting pregnancies. And we have seen from 1985, when we started doing in vivo embryo production and transfer, the model that we had adopted that time was going to remote villages and doing 1 or 2 transfers at the doorsteps of the poorest of poor farmers. I think that model is not going to work in future also. So what we need to have is an infrastructure where we create large or at least medium-sized recipient farms where embryo transfers can be done on a large scale. Because I have seen in US and in Brazil, it is very common that on these big farms, 200, 300 recipients are synchronized at a time, and people just go there and do 300 transfers in a day. That is something that we cannot even dream of in India. So we need to develop our infrastructure in that direction. That is very sure. And secondly, I would have just a very small comment. I mean, of course, after Dr. Baruselli's illustrious presentation, I mean, I cannot comment anything. But as far as the Bos indicus breeds are concerned, I agree that yes, they have a higher number of follicular pool. They produce more number of oocytes, that is true. But I think there is a breed to breed variation also, because now I have worked with 3-4 different breeds, indigenous breeds, and there is definitely a breed to breed variation. Again, within the breed, there is donor to donor variation. And so it is very, very important that we identify the donors with good number of follicular population. Again, there is very interesting observation that there are several donors which produce large number of oocytes, but they don't convert those oocytes into large number of embryos. And then there are some donors which produce large number of oocytes, which also ultimately end up in large number of embryos. So identifying proper breeds, identifying proper donors within the breeds, and finding the best donor and sire combination that produces the maximum number of embryos is the key for producing large number of embryos. I think I would just stop here and let other speakers or other people comment.
Thank you, Sanjeev. Thank you, Sanjeev. Now I just quickly want to ask Dr. Amol Sahare if he's there, if you want to, can take about 2-3 minutes, and if you want to put any comments before we move to the question answers. He's one of our core committee member. Dr. Amol, please go ahead.
Hi, sir. Good morning to Dr. Baruselli and Mishra sir and Kunjit sir. Sir, I want to comment on that only. Sir, it's a nice presentation by the Dr. Baruselli, huge experience in Buffalo, and he illustrated nicely comparison between Bos indicus, Bos taurus, and Buffalo. And I like the study, especially laparoscopic study. He got calves from the calf donor, and stimulation study in post-estrus, especially in Buffalo, is a good study. And also we follow that in I am in JK, and follow that we follow same protocol and same production in bunny breeds. And also we got that. And after a long time, nice to hear Dr. Mishra sir also, and Indra sir also. Thank you very much, sir, for giving opportunity to talk.
Thank you, Dr. Amol. I request now Dr. Siddharth, who is also another core committee member with us, and he is working for a very important organization, National Dairy Development Board. Dr. Siddharth, you have about 2-3 minutes with you. So kindly be in with that restricted time, please.
Yes. Thank you, sir. Thank you, Dr. Pietro, for a very nice and informative presentation. We are working in NDDB for last 3-4 years in embryo transfer, particularly in IVF. I'd like to put one particular model we are working. Dr. Mishra told that getting recipient is very difficult, and we agree to that. So what we created a model is that we started collaborating with the cooperatives. So every organization now in India want to open a IVF lab. So what we told them, you don't open an IVF lab, you use my lab for production of embryos, you bring me the oocytes, I will produce embryos for you, and I'll come down to your union and we'll transfer embryos in union. Only thing you do is to identify the recipients in your union. So what we are doing, we are identifying a central location in every union or every village for that matter. And then they're bringing the animals to that particular location like 3-4 hours before. We are doing the transfer, like not like hundreds or something, but 15-20 transfers in one village or one location. And then we're telling that you keep the animals there for like the day or till evening and then take back the animals. So that model, we call it a hub and spoke model that we are working for last 1 year. And in Banas, we have got success, and in Amul, we got success on that particular model. Now we are moving forward with Mehsana and Savar Union. Another thing, we started working in Buffalo. I like to inform Dr. Pietro that what we are getting is that we don't have very large number of to say now, but we did around 30 OPUs so far. And what we've seen that we are getting around 10 to 12 oocytes per OPU and around 2.3 to 2.6 embryos. But the best part is that we are using a stimulation, only then we are getting this rate. Otherwise, the embryo production rate is very low, like 0.6 or 0.7 it's coming. But when we are stimulating with FSH, then we are getting something around 2.3 to 2.9. But recently in the latest individual, we are getting something around 3. So that is quite encouraging for us. Like today we got from 16 oocytes, we got 8 embryos. 16 oocytes we started in IVC and we got 8 embryos from that. So that's very encouraging for us. But we'll keep in touch with you, sir, so that we can learn from you. Thank you, sir.
Congrats, Dr. Siddharth, for your progress into the Buffalo IVF program. And I don't know if our another last colleague of the core committee is Dr. Ajay Aswal, if he's there. I doubt whether, Ajay, are you there?
I think he's not there, sir.
Okay, sorry. So now, thanks once again to all the speakers. Now, sir, we will take some of more time of yours for question and answers, and we will request now Professor Pietro Baruselli to start with the question. I'll tell you the question answers, and the first question which Professor Misra has asked is about this AMU, antimalarial hormone. If you would like to comment on that, sir, and then we move to the next question. Few more questions, please. Over to you, sir.
Okay, thank you. So congratulations for a lot of research in India that are doing a very good job. I will comment about MEH, but I think that we have a strong possibility here using technology, using embryo transfer, because as you know, in buffalo and in cattle, the interval between generations is so long, it's 4 years, 5 years. So if you use technology to improve the genetic and to reduce the intercalve interval, we can accelerate the genetic gain of our herds. This is a problem in Brazil, probably, I'm sure that's a problem in the world. Huh? So we need to use technology because we need to improve, accelerate the problem. Because it's so long to improve the efficiency of the buffalo and the dairy, the buffalo and the cattle. So this is the concept of the system. But Misra, so the AMH is, as we discussed, is a biomarker, but we cannot, it's difficult to manipulate the AMH. The AMH normally they have a low variation according to the estrous cycle, according to the time. Normally, cows that have high AMH, they have high AMH through the time, low AMH through the time. But it could be a possibility because the AMH is a hormone that control the primordial follicle to start development. So if you remove the AMH at this at the beginning of the folliculogenesis, the animal will recruit more follicle. So one possibility in the future is block IMH to improve the number of follicles during the OPU or during the supermunch. But this is in future studies there, because the MEH could be removed, like some technology, some antibody or some technology to control the MEH to recruit more follicle at the beginning. But we don't have at the moment this technology. So the slow variation, and normally the high MEH is high follicle, low MEH low follicle, but we cannot control today the concentration of MEH in the animal.
Thank you very much.
Probably this is the question.
Thank you,
so,
sir.
But another, okay.
Sorry, go ahead, sir. Sorry.
No, another important discussion that you introduced in the presentation is regarding to recipients. Recipients is a big problem in embryo transfer program. Here in Brazil, the most of the problem today is have a good recipient to receive a large amount of embryos that IVF labs produce. So we are using here in Brazil, we transfer in Brazil around 700,000 embryo per year. So 99% is transferred using fixed time embryo transfer. We use protocols for synchronize the recipient to transfer the embryo in fixed time. So we produce the IVF embryo, we synchronize the recipient, we can transfer fresh embryo because we control the estrous cycle of the recipient, or cryopreservated embryo, depend of the management. So this is very important. Just to understand, 100 recipients, we can have in Neon 8 good recipients using fixed time AI in buffalo and in cattle. So 100 recipients, at the moment of the check of the corpus luteum, we have in Neon 8 good recipients. Of course, this depends of management, depends of nutrition, depends of several things. So this is good because if we expect to produce 8 embryo, we synchronize 100 recipients, and we have at the same time we transfer 8 embryo in the same day. This is very important. I suggest you to study the possibility you have in India to synchronize a large number of recipients to improve the IVF system. And of course, the hormones that we have here in Brazil for synchronization is different than the hormone that you have in India. So we can use here estradiol, we can use PMSG or ECG. You probably cannot use estradiol, I don't know, but I think that in India you cannot use estradiol. But here in Brazil, we can use it. So it works very well, the program using progesterone, estradiol, PMSG or ECG, and prostaglandin. This is very important to support the IVF industry, to improve the number of good recipients that can receive a IVF image. Okay?
Thank you, sir. Thank you. Now, one question is there. There are few questions are there. We request for your some more time of yours. And Dr. Amol is asking, according to follicular wave study, what should be the interval between the 2 OPU to get better recovery rate? What should be the interval? And second question is that when you are doing OPU with a non-stimulated donor, what day we have to do OPU post-estrus? These are the 2 questions.
Good question. So the interval between OPU, we did a study that depends of the amount of the follicles of the donor. The donor that have a lot of follicle, we need to increase the interval between OPU. The donor that we don't have a lot of follicle, we can decrease the interval between OPU. Why? Because the donor that have a lot of follicle, we have more damage in the ovary doing the OPU. So we need more time to wait for the next OPU. Like today in Zebu in Brazil, we are using 21 days interval, 21 days. But in Holstein, we can use it once a week or twice a week, depend of the amount of the follicle, once a week or once 14 days. So the interval in Buffalo, we are using each 14 days, one OPU, because they don't have a lot of follicle. They have the damage of the ovary is not so strong. We can reduce the interval. So depend. So today, normally in Zebu is 21 day, and in Buffalo is 14 day. And if you need to decrease, could be, but when we decrease, the second OPU decrease the efficiency. Okay, I don't know if I can.
Yeah, no,
my English is not so good.
No, sir, it is very, very clear. You are very clear, and you know, our own experience is that looking at the literature, we started doing OPU twice in a week, every Monday and Thursday. But this is a very important learning from your observation, is that the animals with more follicles, your frequency should be at least 21 days. What we did is that we found some donors, they are extremely good with the follicles, and we started aspirating them even twice a week, which ultimately resulted into, I mean, over a period of time, the oocytes went on decreasing and the ovaries started becoming hard. So that was a very right, you rightly said that we should have a interval between 2 up to 21 days where we have a large follicular numbers. Thank you, sir. There is a question from Sanjeev Chaubal. After how many OPU you start seeing a decline in oocyte recovery in Bos indicus donors? I mean,
2 or 3 OPU, they start to decrease. So normally the first OPU, we have a high amount of oocyte, and so they decrease until the second and third OPU, they decrease the oocyte production. After they maintain stable until the end of the program.
No, with this, I want to ask you, sir, if you keep an interval, say 21 days into Bos indicus, and they are prolific oocyte producers, can you, prove to be a good donor? Can you continue for 2, 3, 4 years without any problem?
Yes. We today the industry in Brazil, they select the genomic donors. They put this high genomic Bos indicus or Bos taurus in the AI embryo transfer center, and they are doing OPU for 1 year, 2 years, continual OPU for 1 year, 2 years. Depending of the donors, they produce normally. Some donor at the end start to decrease the quality, and so or they become pregnant or they remove from the embryo transfer program. But it's possible do OPU for 1 year, 2 years, dependent of the donor. Normally, 60% of the donor produce through the 1 or 2 years. Some donors start to decrease, and so we need to change, or we become them pregnant, and the next year we start again to collect the oocyte. Because we have a metabolic problem, it's a little difficult to need more time to discuss with you. But the donor that become aspirated, they is not pregnant. So they become fat, and they start to improve insulin resistance. The fat animals, I did this study here in Brazil from insulin resistance. So the insulin resistance try a metabolic problem to introduce the glucose on the oocyte. So the fat animals that have insulin resistance, decrease the oocyte quality because they have a low apport of glucose in the oocytes. So some problems of continuous OPU is the donor become fat and decreasing the oocyte quality on the time. So we need to control the nutrition, but if you have a good control of the nutrition, you can do for 1 year, 2 years OPU without a problem.
Thank you, sir. You briefed us about the aspiration through the laparoscopy of the young calves. I would like to share our experience that we tried about 9 to 12-month-old heifers, prepubertal heifers, and we could aspirate and get pregnancies from them. Only thing is that we have to choose our OPU technician who has got a smaller hand so that he can put a hand into the rectum, and we definitely use that heifer probe developed by WTA, and it worked very well. But definitely some of our universities can take the trials with this laparoscopy also for production of the calves. There's one more question from Dr. Chauvel again. Do you use Ghirlando as a donors? If yes, how is their performance?
It's good. It's bad. So we use Ghirlando. The performance of the Ghirlando is better than hosting, than hosting. So the Ghirlando produce a lot amount of oocyte, good oocyte, and a lot of amount of embryos comparing to hosting. So works very well the IVF in Ghirlando here in Brazil.
Thank you. This is a question from Dr. Sanket Patil. He is asking, talking about endocrine profile, does endocrine P4 levels have any correlation with embryo production?
Sorry, the question is progesterone?
Yeah, progesterone, right. P4 levels have any correlation with embryo production?
In IVF, we did a study. We block with generate agonist. We block the donors. We remove the cyclicity, and the control group we have cycling animals with progesterone. No, we didn't found after 6 months of OPU, didn't found any effect of progesterone on oocyte quality. So respond, we are not able to understand that the effect of progesterone, and we didn't found any effect. We didn't found any effect of progesterone and oocyte quality and IVF efficiency. So no effect of progesterone on oocyte quality we found in our study.
Thank you very much, sir. And there are a few more questions, but we already taken your 2 hours. And so for the rest of the questions, what I will do is that email to you, and then we can seek answers from you. And now, so at the end of this question answer session, it comes to an end. On behalf of all the members of the Bovine Embryo Transfer Group of India, I express my sincere thanks to you, sir, for covering various aspects of the bovine IVF and embryo transfer technology. And I also take this opportunity to thank Professor A.K. Misra and Dr. Indrajit Singh for making themselves available for this webinar. I'm also thankful to all the members of Bovine Embryo Transfer Group who are attending this webinar, and all other participants from various parts of the world who are attending the webinar through the Facebook Live. So with that, sir, we again express our deep gratitude and thank to you. Once again, I repeat my request is to kindly look at the some collaboration with some universities and national organization for taking up this work further to different level, because I understand government of India has got some agreement with the Brazilian government. So under that, whether we can get in that agreement, whether we can get some of your faculty to give a training to our people, or we can take some collaborative project. So Professor Pietro, I again request you to look into that as a national mentor. This is my sincere request to you. And once again, thanks everybody. And I declare this session as closed. Thank you very much. Thank you, sir. Namaste.